in the chat box in a second. So at the end of the seminar, as usual, we will have a Q&A session. Please feel free to post your questions during the talk in the tab Ask a Question. And finally, I would like to encourage everyone which is interested in getting to know Marija in more depth, or if you want to go in a broader discussion after the talk, just join us. We will have our usual meeting on the Zoom room after the talk. So I will share the link in a second. And now, without really any further ado, I would like to introduce you to our today's speaker, Professor Marija Kundakovic. She received her PhD in biochemistry and molecular genetics from the University of Illinois at Chicago, and she completed her postdoc training at Columbia University and the Icahn School of Medicine at Mount Sinai in New York City. In 2015, she was awarded an Arsad Young Investigator Awards by the Brain and Behavior Research Foundation, and she established her research lab and is currently an associate professor in the Department of Biological Sciences at Fordham University. Dr. Kundakovic has been at the forefront of psychiatric epigenetics research since her early career, and her lab now focuses on hormonal and environmental factors which drive sex differences in depression, anxiety, and substance use disorders. Her lab discovered sex-specific epigenetics regulation in the female brain as a function of the ovarian cycle, providing a new molecular framework to study the female-specific susceptibility to psychiatric disorders. And I found this to be a really fascinating field. And so I would like today to welcome Marija at our series, and I'm really looking forward to hearing from you today. So the stage is now yours, and I invite you to share your presentation.
First of all, thank you very much for this generous introduction and also for this general invitation. So let me just make sure. Can you see my screen? Hello?
Yes,
are you able?
It's perfect, okay. Marija. Yes.
Okay, so you're able to see my screen. I just want to make sure
that I can. Yes.
Going. Okay, perfect.
Everything is
okay. Yes. So we are ready to start. So first of all, really, thank you so much, Dita, for this generous introduction and for this invitation. I feel very honored to be here and to share this with this audience. We just actually had a very short, conversation before, before starting this session that this is a very special audience that actually has appreciation to what something outside of the brain can do to the brain. And I think 2 weeks ago you had this presentation with Emily Jacobs about how the sex hormones can affect the brain in humans. So I think that was a good introduction in what I will be talking about, but my work is much more molecular, right? So it actually focuses on the molecular mechanism underlying this. But I wanted to really start with a bigger picture, why I am actually doing what I'm doing, right? So when I started my career, really, I'm a molecular biologist, I'm a neuroepigeneticist, and I really wanted to try to contribute to this understanding of the molecular basis of psychiatric disorders, right? So that was something that's how I started actually being involved into epigenetics research. And my lab is primarily focused on anxiety and depression, right? And we know that these disorders are very complex, right? We know that they run in families, so genetic factors are playing a really important role in these disorders. We also know that they are having this environmental component, right? That particularly stress is important because that can precipitate your depression episode or something. And the idea of this field, when the field actually developed, which was let's say 20 years ago, is the first time that neuroepigenetics field actually started, particularly when we talk about psychiatric epigenetics. The epigenome was something that now we have as a biological substrate through which stress and genes can interact to change gene expression, to change brain structure and function, and contribute to anxiety and depression symptoms. Of course, this is very simplified, as you can know, but it's really encouraging. We have a lot of studies showing, for instance, in animals, that stress can change the epigenome, that this can change behavior. We also have studies in humans that actually are showing that there are epigenomic changes across different psychiatric disorders. However, one of the things that really hasn't been addressed so much is that these disorders are actually quite sex biased, right? So when we talk about anxiety and depression, these disorders are at least twice as prevalent in women than men. And when you think about it, there has to be like a biological, etiological factor that actually contributes to this. And there has to be a mechanism that actually can explain these differences, right? And unfortunately, this hasn't been done enough because the majority of studies, in neurosciences and in neuroepigenetics were done only in males, right? So what I would like to invite you for today, if we can consider the sex hormone status together with genetics and stress, and how this can change the epigenome and change gene expression to contribute to changes in brain structure and function, anxiety, and depression symptomatology, right? And I want to explain to you why I believe that ovarian hormone status is particularly important, right? So I like starting with this particular graph. And this graph actually shows how the depression risk, this is the upper panel, shows how the depression risk changes in women and men. Women are shown in the purple and men in blue. And then how this actually corresponds with the changes in sex hormones in females, right? So what I would like you to see here is that basically, if you take a look very early in life, like pre-puberty, right? The risk for depression in women and men is very similar in boys and girls, right? So the risk really jumps to 2 times higher in girls once they start having their first menarche, the first period, and when they start fluctuating with hormones. Then throughout the reproductive years, we have actually 2 times higher risk for women than men. And this corresponds to this really fluctuation in hormones. And then finally, in the perimenopause, you actually have a further jump in risk that is associated with the most erratic hormonal changes. And then postmenopause, when you have actually this decrease in sex hormone status in women, interestingly, the risk becomes very similar to men, right? So what I would like to take you from this particular slide is when we are talking about these sex differences in depression risk, it's really not high or low ovarian hormone, but really these fluctuations in hormones that somehow seems to introduce certain vulnerability in women, and this seems to somehow contribute to this vulnerability and increased risk. So, and we would like to understand the molecular basis for this, and this is what I would like to talk about today. So why this is important, why estrogen can actually affect the risk for depression, right? So as I told you, you already know from the, if you've seen the speaker 2 weeks ago, that estrogen is actually a potent neuromodulator, right? So people talk, when they think about estrogen, they think typically about reproductive hormone, right? But estrogen is really a potent neuromodulator. And particularly when we think about systems such as serotonergic and noradrenergic system, which are actually very important for emotion regulation and for, these are the targets of actual anxiolytic and antidepressant drug. So what we can really say is that estrogen can affect this system. However, what is really interesting is that estrogen will increase serotonergic function. It will increase noradrenergic function. So it's important here to say that estrogen actually has something that can be seen as an anxiolytic or antidepressant-like activity. And estrogen will even increase BDNF levels, right? So now you may ask the question, if estrogen can something like antidepressant-like activity, why would women be at the higher risk than men, right? And I think the key is actually that in fluctuations, right? Because if you had maybe the stable estrogen levels, that would actually be very beneficial. But if you had this fluctuation, there is always this withdrawal or drop in estrogen, which requires periodic neurochemical adaptation. And this is something that I think increases the anxiety or depression risk. And this is, again, something that we would like to understand at the molecular level. And because I'm molecular scientist, I'm always thinking about mechanisms, right? And estradiol specifically, I think the group here knows that estradiol can actually work through both nuclear or membrane-bound receptors, right? So if we are talking about nuclear receptor, estradiol will get into the cell, bind to estrogen receptor alpha and beta, and then bind to the genomic region that we call estrogen response element, and then will change gene transcription, right? Another possibility is that estradiol can bind to the membrane-bound receptors, activate certain kinases, and then one of the possibilities also that it can phosphorylate the transcription factor, and then transcription factor will go, and again, you will have indirect genomic effects, right? So this is something that I'm going to bring up several times in my presentation because as we are going through our data. So the question of mine was, because I'm a neuroepigeneticist, are there chromatin regulation that is actually induced here in response to these changes in hormones that we are seeing? And now that I'm invited to give this talk, it's obviously that we've been successful to show this. But what I really would like to know, when we were starting, the only evidence that we had is that, chromatin can change in response to estrogen in breast cancer cell lines, right? And it's very different to talk about how hormones can affect dividing cells, right? And post-mitotic cells such as neurons, which we were actually interested in. So just to give you the idea about the epigenetic regulation, when we talk about epigenetic regulation, particularly, we are interested in how chromatin is regulated, because we know that in our cells, DNA is not naked. It's actually packaged into this very specialized structure that is chromatin, called chromatin, where you have the DNA that is wrapped around this histone protein, right? And what is important is that, like, you know, when chromatin is in this closed orientation, let's say it's highly methylated, you have it closed, right? So that it cannot, transcription factor cannot bind, and you cannot transcribe the genes. However, if you open chromatin, right, the transcription factor can bind, and then you can actually have active gene transcription. So we are interested in how, in these dynamic changes in chromatin opening and closing that can actually control gene expression. And, you know, particularly, as I said, like, we're interested in how this is happening post-mitotic neurons in response to hormonal changes, right? And again, I'm going to say again, I think when I was starting, like, my lab, that was like, you know, 7-8 years ago, this seemed like a very risky project, but I'm really happy that we actually pursue this project, because I think, as Zita already said, like, you know, it really generated some very exciting data, which I'm happy to share with you today. So what we are doing, ideally, you would do in humans. And I really want to be inclusive here and say that what we do is actually applicable to all menstruating individuals, right? So you don't have to be cisgender woman for this to be applicable for you. If you're a non-binary individual or you're a transgender man who menstruates, our data are going to also be applicable. So this is kind of a gender independent. It's more related to actually changes in hormones that if you have ovaries, right? That's something that I think it's always important to emphasize. So if you study this in menstruating individuals, you know, like, I think pretty much everyone should know here that, like, you know, we have the follicular phase when there is like a high estrogen and low progesterone, and we have a luteal phase when there is a low estradiol and higher progesterone. So because it's really difficult to study this in humans because the brains are not accessible, we study this in mice. And mice have something that is very similar. It's called the estrous cycle. It's shorter, of course, 4 to 5 days, but the actual hormonal profile is very similar. And we typically focus on 2 extreme phases: proestrus, when you have high estradiol and low progesterone, which is mimicking the follicular phase of menstruating individuals. And we focus on diestrus, which has a low estradiol and high progesterone, which is mimicking the luteal phase of the menstruating humans. So the way how we do it, we do this very comprehensively. So we will take the vaginal smears daily for at least 3 consecutive cycles, or let's say 2 weeks or so. And then based on the smears, we can actually say whether someone is in proestrus or diestrus or any of these groups, right? And I also want to say, I'm not showing this data because, like, you know, like, we actually do this now non-invasively, but we did also check the hormonal levels in serum to just confirm that what we really see under the microscope as a proestrus is really hormonally what we expected to be high estrogenic and low progesterone, right? So after this comprehensive tracking of the cycle for 3 cycles, we then placed animals into either diestrus or proestrus group, and then we always test them with age-matched males. And this is basically our study design that I would like to start with. So these studies were started with my first postdoc, Ivana Jaric, and then this was actually the majority of the actual research that I'm going to show was done by my graduate student, Devin Rocks. And then we also started this with our, you know, like, dear collaborators, John Greeley and Masako Suzuki from Einstein School of Medicine and, you know, Texas A&M University. So the way how we do it, again, we have the 3 different groups: diestrus, proestrus, females and males. I'm always going to use the same color scheme. And then I'm going to show you first some behavioral data, just so, you know, you can see why this is important behaviorally speaking. And then we are going to go into the molecular stuff that I'm primarily excited about. So we use this very simple anxiety-related test in mice, right? I think probably the audience knows, but, you know, these are short tests. So open field LiDAR box and elevated plasmase. So in order for the mice to say that they're, you know, like, anxious, or they are the way how they typically behave, mice actually prefer to be around the corners, in the dark areas and in the closed areas. However, if you see less anxiety, that means that the mice will be actually spending more time in the center, in the lit area, and in the open area. And for instance, if you were to give an anxiolytic agent, this is what mice would actually start spending more time in the lit and open areas, right? And the reason I want to say and address this at the very beginning, we know that these tests are very simple. People would ask us, but this is not really anxiety. But these tests are, first of all, I'm going to explain why this is translationally relevant, or at least I believe that this is translationally relevant. And then it is really important for us to have this really simple test that we can do in 5 minutes. The reason for that is because the cycle changes all the time, and you really want to have like some good test that can tell you in certain stage how the behavior of the animal changes. So this is why we actually really prefer to have this simple test that we are doing. So, and I would like to actually show you some example of the elevated plasmase. This is high estrogenic females, low estrogenic females, and male. And the open area, the open arm is actually this one. So it actually corresponds to this. And if you look at this like long enough, you will see how these high estrogenic females actually spend a lot of times in this open arm. So she's fearless, she goes around, it's like a fashion week for her. Well, if you look at these low estrogenic females, you see how she's very confined to these like, closed arms, and males would be somewhere actually in between, right? So, and we see this same behavior across the 3 different tests, right? So we always see that high estrogenic females are behaving more similar to males. And whenever there's a drop in estrogen, this is when we see this increased anxiety indices. And again, I want to refer to the human, studies that actually show premenstrually, for instance, you are seeing also this drop in hormones, and you're seeing increase in depression symptomatology, for instance. Or if we are talking about postpartum depression or perimenopausal depression, we are seeing whenever there is like this drop in estrogen or drop in ovarian hormones, this is when we are seeing this actually increase in anxiety and depression symptomatology. And this is why I believe this is really like a good model that has a constructive validity to actually study something that is anxiety related. But we wanted to actually understand more these behaviors, right? And I also want to emphasize here, because oftentimes you hear me say estrogen, but I do want to say that estrocycle is not only estrogen, right? There is progesterone, there's a lot of going on. But we do have evidence from our labs showing that the behavior that we are looking at, elevated plasmase in particular, is actually primarily estrogen driven in our mice, right? So when Devin, actually my graduate student, joined the lab, the first thing that he did, he wanted to actually check the elevated plasmase behavior across the cycle, right? So I typically show you the data only on proestrus and diestrus, but he looked at all these different phases. And this is like how the behavioral analysis looks like, and this is like how your estradiol levels look like. So we actually see that our behaviors are directly following the estrogen curve, right? The higher the estrogen, then less the anxiety you get. So Devin then decided to actually check this by injecting exogenous estradiol. So what he did, he took these animals that have very low endogenous estrogen levels, and then he injected them with estrogen, and immediately after 4 hours, he was already able to see that there is less anxiety indices that you are seeing. So basically, by giving exogenous estradiol, you can basically mimic what we are seeing in proestrus. And so my graduate student, Layla, then what she did, we were thinking, but what if you do ovarectomy? You remove ovaries, and then you try to add estrogen. And you can do the same thing. You can actually also decrease anxiety, but you need the multiple injections. You cannot do it very fast. And I'm going to go back to this, because if you do ovarectomy, we are seeing that there are a lot of brain adaptations. So it seems like the brain needs to reprogram itself to be responsive to hormones again. So now we saw all of these changes in behavior across the cycle, and specifically in response to estrogen withdrawal. And now the question was like, are there any structural changes? And I like actually showing this because, like, people always get very, excited about this data, because it's almost, it's really remarkable that you, across maybe 24-hour time scale, you can see changes in dendritic spine density across the cycle. And like, so for instance, during the high estrogenic phase, you see increase in the dendritic spines, and then you see the decrease in the dendritic spines during the low estrogenic phase. And this is really important, because for me, this represents in a way the structural substrate for this increased vulnerability for females. Because if you in males do, expose males to stress, you will see decrease in the spines. If you expose males to ketamine, for instance, you will see increase in the spine. So what I think our studies are actually showing is that estrogen is a neuroplastic agent. But whenever you have this drop in estrogen, this is when you are seeing this lower dendritic spines and increased vulnerability, right? These studies actually were previously in 90s actually done in Bruce McHugh's lab, but in the dorsal hippocampus. What we are showing now is important because it is in ventral hippocampus. And I just wanted to very briefly explain why we are focusing on the ventral hippocampus, because you could potentially look at many different brain areas that can be of interest for anxiety-related behavior. So there was this, I would say, a really robust body of evidence from both earlier study where people would do lesions of the ventral hippocampus, or more modern study where particularly from Renee Hand's lab at Columbia University, that were showing if you optogenetically activate ventral hippocampus, you are going to see changes in anxiety-related behaviors, but not in the spatial memory. And the opposite, if you do that in the dorsal hippocampus, you will see changes in memory and not in the anxiety-related behavior. So we thought this is really like a good region. It's like a subcortical. It's like quite well preserved across the species. And we know a very good evidence that these neurons in this area are actually driving those behaviors that we are interested in. So we were thinking, the first hypothesis is like, whether these changes in the hormone that we are seeing across the cycle could possibly open and closing chromatin in these neurons of the ventral hippocampus and change gene expression so that this can actually, at least in part, underlie the behavioral and structural changes that we are seeing. So that was our starting hypothesis, right? And the way how we look at the chromatin, we do that with a neuronal specific ataxic assay. And so let me just explain what this chromatin accessibility assay is all about for people that may not actually be very familiar with. So what we do, we have our three groups, diestrus and proestrus, females and males. We take the ventral hippocampus, then we sort the nuclei, and then we specifically sort the new N positive nuclei. So new N is a marker of postmitotic neurons. So what we do this, we are sorting to actually have a higher resolution of the assay, because as you probably know, glia cells and neuronal cells have very different epigenomes. So we wanted to increase the resolution of the assay. And the way how this works, there is an enzyme that is called the transposase, which will cut the DNA only when chromatin is open, right? So you are basically enriching for open chromatin regions. And so when it cuts the DNA, it will also induce, and incorporate the sequencing adapters. So you can do the PCR and sequencing and then do bioinformatics. And whenever you see this open, like a peak in the data, you see that the chromatin was open. When you see no peak, that means the chromatin was closed, right? So the first question that we had was if you take these diestrus, proestrus, and males groups, and you isolate these ventral hippocampal neurons and do this ataxic assay, how many open regions do you have, and how different they are between each other? And importantly, the number of open regions was around 60,000, right? So they were similar. But what really we found was really striking, right? We found that around the 30% of the genomic region in ventral hippocampal neurons changes chromatin organization across the estrous cycle. This was really not the degree of change that we actually expected, particularly because what we are doing is physiology, right? What people would do typically, they would actually do the overoctomy, add estrogen, but we are showing in this physiological model like a huge reorganization of chromatin across the cycle. What is also very interesting is that the degree of changes that we are seeing across the cycle is very similar when we compare diestrus to proestrus and when we compare this to diestrus and male or proestrus and male. So the within female variability is very similar to between sex variability. And this is something that we also did not expect. So we did a lot of bioinformatics analysis, and I'm just kind of focused on important points here. So the first question was, what are the genes that are being affected by these chromatin changes? And importantly, when we did the gene ontology analysis, the main term that we were getting was modulation of synaptic transmission, regulation of membrane potential, synapse, and so on and so forth. So what we are seeing is that the neuronally relevant genes, the genes important for neuronal function are being, we are changing, the chromatin is changing around these genes, basically. And here I'm just showing you, sorry about that. Here I'm just showing you some example. This is, for instance, the gene that is encoding the subunit of AMP receptor. This is the potassium channel gene. This is synapsin-1. And just to give you example, for instance, when you have high estradiol, for instance, for the GRIA3, you have this opening of the chromatin when the estrogen is high, and then there is a closing of chromatin when estrogen is low. And this is associated with changes in gene expression, right? So two things that we can take from this particular slide. First, that chromatin changes are happening around the genes that are very important for your neuronal function. Number two, that this is also associated with changes in gene expression, which means that this is functionally relevant. So then we did another type of analysis. For instance, we did pathway analysis, and we then asked a very specific question, right? So we asked the question, if you take all the regions that are specifically open during diestrus and all regions that are specifically open during proestrus, you merge them and you do pathway analysis. What are the pathways that are showing the same pattern that we are seeing in our behavioral data? And just to remind you, what we are seeing in behavior is that high estrogenic females are more similar to males, while basically the low estrogenic females are different from both of these groups, right? And there were only two pathways that show the same pattern in our chromatin data. And these are serotonergic synapse pathway and hedgehog signaling pathway. So we are going back to serotonergic synapse. As I already told you, number one, estrogen was shown previously to change serotonergic function. We know the serotonergic function is very important for emotion regulation. The main SSRIs as drugs are actually affecting this type of neurotransmission. So what we are now providing for the first time is the molecular mechanism to how estrogen and other ovarian hormones can change the serotonergic function, right? And just to give you an example of how this is working, for instance, this is the gene encoding serotonin receptor 2 beta. So you see more open chromatin during low estrogenic phase and more closed chromatin during the high estrogenic phase, and this is actually associated with changes in gene expression, right? So this is like, an example how differential chromatin organization within serotonergic genes can provide the molecular mechanism for sex-dependent variation in anxiety-related behavior, right? So now I already told you we could potentially also look at and ask the question, is this going through the nuclear bound or membrane bound signaling pathway, right? So basically the question that we can have is like, let's say if we take a high estrogenic phase of the cycle and we look at which regions in the chromatin are specifically open during high estrogenic phase of the cycle, are we seeing the enrichment of estrogen response elements, or we are seeing the enrichment of something else, right? And interestingly, so this is exactly what we did, right? We took all of these, pro-estro specific region. We did a motif analysis asking the question, which is the transcription factor that bind to this a region that are specifically open during high estrogenic phase. And interestingly, we did not find the enrichment for estrogen response element. We found the enrichment of this, MEF2 proteins, but one of the actually binding motifs was basically very intriguing to us. And that was like one for the EGR1 transcription factor. So there are at least two reasons why we were excited about that. First of all, EGR1 is estrogen responsive immediate early gene, right? So what this means is not that only you see opening of the binding site for EGR1 during the high estrogenic phase, but you see induced expression, right? The mRNA level of this gene are also induced when you have high estrogen. Another thing why we were actually excited is because it's an immediate early gene product. And there were studies from Mike Greenberg at Harvard and also from Zhong and UPenn that actually showed that these immediate early gene products can actually be chromatin regulated, that they can be opening chromatin, right? So, and this is why I'm going to go at the end of my presentation, I'm going to go to this EGR1 because we functionally manipulated this specific transcription factor to show it's important. But for now, I just wanted to say that, like, our data are more consistent with the membrane bound pathway, where we believe that actually ER alpha will induce, like membrane bound ER alpha will induce something like EGR1, and it will bind and open the chromatin and change gene expression. And we did a confocal analysis to actually confirm that indeed in our ventral hippocampal neurons, we indeed see the membrane-bound estrogen receptor that are here co-localized with our DARPP32 is one of our target candidate genes, and then MEP2 is actually neuronal marker. Okay, so then another question that we had, like I already showed you that our chromatin changes correlated with some changes in gene expression, but we were doing this only for certain candidate genes. And now we wanted to know how many genes actually change expression in ventral hippocampal neurons across the cycle, doing RNA-seq. So what we did, we again took our three groups. We took the ventral hippocampus, did a nuclei sorting, specifically isolated neuronal or neuron positive nuclei. And then instead of doing a TAC, we did actually RNA isolation and we did RNA-seq. And we found around 172 differential expressed genes in the proestro to diestrose comparison. And the question like what are these genes that are changing expression across the cycle actually? What are these genes in general? What do they do? And, of course, the genes actually are very important for neuronal function. And specifically, we like this idea that one of the terms that we got was behavioral fear response, which actually is very consistent with our behavioral data that we are seeing changes across the cycle. But there was something that we did not expect at all. And that is that basically there were this set of genes that were changing the expression. And these genes actually encode the ATP-dependent chromatin remodeling factors. So when you see chromatin, like, in order to open or close chromatin, you need some of those chromatin remodeling factors that will kind of start shifting, like, the histones and, like, start, and use ATP energy to actually open the chromatin, right? So what we are basically seeing is not only that there are changes in chromatin organization across the cycle, but there is the change in the expression of the chromatin remodeling factors across the cycles, which tells us that there's really something about the regulation of chromatin that is really important for the regulation of neuronal function across the estro cycle. So when we did the overlap between ataxic and nuclear RNA-seq data, we again saw the main cluster was this chromatin remodeling cluster. And when we did the motif analysis, we saw that basically the main motif, again, one of the main motifs was EGR1. And this actually told us that EGR1 is an important regulator of chromatin and transcriptional changes. So now I want to keep you focused on behavior, right? Because I told you what we are doing is like we want to understand how behavior changes across the cycle because we want to have this applicable to the anxiety and depression disorders, right? And so I just want to give you my, like kind of a framework that, or hypothesis that I like to work on. And I'm just going to use a single gene because it's got for the simplicity, right? So just to explain to you how I think these changes that we are seeing, the molecular changes, can actually be involved in these behavioral changes that we are seeing in females across the cycle. So for instance, DLK1 is one of these very interesting imprinted genes that's been shown by another group that were doing things only in males, right? If you knock down the DLK1 in males, you are seeing increase in anxiety. In all these tests that I showed you are changing across the cycle. The point that I want to make here that while in males you need to do the knockout, in females you have the natural variation, right? So you don't need to manipulate females to increase anxiety behavior. Whenever you have this drop in estrogen, this is when you are seeing actually decreased anxiety. So my point here is that, like, if we have these molecular changes, such as changes in DLK1 gene, where with the decreased estrogen, there will be increased expression of this gene, right? That this by itself can actually induce this behavioral or psychiatric risk that we are seeing, right? And how is this actually connected to chromatin? It is connected to chromatin because this increased DLK1 expression that we see in hystrogenic phase is also associated with this increased chromatin across in the proestrous phase. And then when you have the low estrogen, you are actually seeing closing of the chromatin. And this is actually happening in the place where the EGR1 motif is. So the idea here is that when you have induction of estrogen, EGR1 will bind to its sites, open the chromatin, increase expression, and decrease anxiety behavior. However, when you have drop in estrogen, you have everything opposite, right? And this is where this vulnerability is coming from at the molecular level. Okay, so what I showed you so far is basically that when you have this, across the cycle, you have this opening and closing of the chromatin that is associated with some partial changes in gene expression, and this is associated with changes in synaptic plasticity and with changes in behavior. However, what I did not tell you is like what you can study with the toxic is actually what we call the 1D chromatin. One chromatin actually in our cells is packaged in this very specialized structure that is called higher order chromatin organization or the 3D genome, right? And so you see all these different levels of organization. And what is important here also to mention is this is like a huge black box in neurosciences, right? So when we started doing this 3D genome studies, basically there was only a couple of studies in the brain and they were all done in male animals, right? So we decided to do a high-C analysis that I'm going to just now briefly explain, and then to basically overlap this data with the toxic and RNA-seq data to understand how these 3D genome changes then can change gene expression across different groups that we are interested in. So again, this was, led by Devin and also done in collaboration with our bioinformatics colleagues, Jordan Rowley and Mamta Shukta at University of Nebraska. So just to briefly explain to you why 3D genome organization studies are really important, right? So when I was telling you about the toxic data, when you see changes in chromatin, we literally assume that, let's say, if chromatin changes you are seeing in certain region, that the first gene that is the closest to the change would actually be something that is affected. However, if you look in our genome, our DNA is actually making all of these DNA loops, right? It's shown here. So something that is really far away in the linear genome can actually be brought together by these loops, right? And this is something that can actually regulate the genes specifically. But the problem is, like, if you do a toxic, you don't have this information, right? So you really have to have a very specific chromatin conformation assay that will actually show you. And we do this in a non-biased way, so we actually profile this throughout the genome. So how we do it, we take our 3 groups we take the ventral hippocampus, we do the nuclear sorting, we sort the neuronal nuclei. And then we do this whole series of steps. I'm not going to go into it, but the whole idea is that you cross-link this, and then you end up in your library in something that was actually closed in the 3D genome, now is actually closed in the linear library, so that you can bioinformatically profile and say, what are all these 3D genome interactions that can potentially affect gene regulation? So the data typically looks something like this. You have all these dots are actually representing some of these interactions, and then you see that there is some kind of pattern here, right? So, and I just wanted to explain to you what we did. We look at the 3 different levels of interaction. So just to give you the idea, if you look in our nucleus, typically each of the chromosome is in a special territory, right? So typically you don't have a lot of interaction between the chromosomes, right? But if you zoom in each chromosome, what you are going to see is something that we, for instance, call compartments, right? This is the first level of organization where you have, for instance, inactive compartment. We also know this as a heterochromatin. And then you have the active compartment that we also know as a euchromatin. And the way how this looks, for instance, this is your active compartment that is associated with this is RNA-seq here at the time. So it's associated with active genes, and then you have inactive compartment associated with no gene expression, and so on and so forth. So you have this pattern that actually looks so now our data looks very nice in terms of this compartmental organization. Then you have the CTCF loops. These are the loops that actually allow for these long range interactions. And again, each of these little dots here will actually represent one of the loops. And then we have enhancer promoter interaction that actually are a little bit more dynamic. So each of these arc here is actually one of those enhancer promoter interactions. And the more of these interactions you have, the higher RNA-seq level. So basically, you can see that with increasing the number of interactions, you are increasing gene expression, which means that they are important for gene regulation. So now what I'm going to show you, if we go to each of our groups, like diestrus, proestrus, and males, how this changes in terms of all these 3 levels of organization across the sex and across the cycle. And when we look at the compartmental differences, the only actually chromosome where we see compartmental changes was X chromosome. So now I have to be very clear here that you absolutely expect to see sex differences in X chromosome compartmental organization, because in males you have only one active X chromosome, while in females you have one active and one inactive X chromosome. And we know from the previous study that inactive X chromosome is very has very closed structure, right? So because female genomic data are going to be the average of the two, even if you are not an expert in 3D genome, you are going to look at this data of diestrus and males and say, oh, they look very different, right? However, there was something that we did not expect. And this is when we start looking at the proestrus, when the estrogen level rise during the estrous cycle, you're starting seeing some of these compartments that actually start to look more like males than like diestrus. And this was really quite specific to the X chromosome. And again, we see a good correlation, like the signal goes very similar in the high-estrogenic phase as it goes in the males when we compare them to diestrus. And there was something really intriguing that we found. And I already told you that, like, chromosomes are actually have a very little interaction between each other. But what we were actually starting to see is that, like, when you start increasing estrogen in the high estrogenic phase of the cycle, it seems that the X chromosome starts having more interaction with other chromosomes. So this basically told us that maybe there is this increased volume of inactive X chromosome during the high estrogenic phase of the cycle. So basically that there is some kind of a decondensation event during the poiestrus. Of course, knowing how close this structure of inactive X chromosome is, this is kind of a radical idea, but as you see, like, we are getting more and more data that actually are showing that there is some dynamism of inactive X chromosome that we are seeing across the cycle that is quite remarkable and very interesting. So in order to actually confirm that there is really a compartmental change in the X chromosome across the cycle, we also did the FISH analysis. So what we basically did, we took our Hi-C data and then we constructed 3 different fluorescent probes. One of the probes would actually switch from active to inactive compartment across the cycle. So what we did, we took the ventral hippocampus, like brain slices, and then we put this fluorescent probe so that we can actually microscopically, at the fluorescent microscope, see the physical differences between these probes, right, to test our hypothesis. And we were actually able to confirm what we were inferring from our 3D genome data. And this is that basically during diestrus, you see these two probes to be very close to each other in the same compartment. However, during proestro, there is like a physical shift in these two probes, and so they become more similar to males, right? So we are basically confirming that there is a 3D genome change in the X chromosome across the cycle. And you may now ask, but is this associated with the gene expression changes? So we are not seeing that many gene expression changes associated with this X chromosome dynamism, but we are, for instance, seeing the change in the gene, such as this gene encoding the serotonin receptor 2C. This is actually one of those SKP genes. So it's more highly expressed in diestrus than in males, but then it becomes actually more similar to males during the high estrogenic phase. So we actually coined this new phrase, estrocycle stage dependent SKP genes. So this seems like there are some genes that might be escaping X inactivation during one phase of the cycle, but then they become more similar to males during this high estrogenic phase, right? So that's really something intriguing and something that we are actually extensively working on, because I think it's one of the most exciting findings from the lab. We looked also at these CTCF loops, found around 600 differential loops. Interestingly, again, when the estrogen rises, they become more similar to males than to low estrogenic females. But now these loops are actually more different in autisms, right? It's not all about sex chromosome. That's something that is actually important. And actually now the differential loops that we are seeing are in the autism, not the sex chromosome. And I'm just now giving you an example of the gene that is encoding PCAP. This gene is important because we know that it regulates emotion it's actually important for emotion regulation in mice. It's also like regulating anxiety-related behaviors in mice. And it's also been shown that this PCAP can be one of the interesting sex-specific markers for the PTSD in humans. So what we are seeing here is that basically we are seeing stronger loop in the high estrogenic phase than in males, and then the loop actually is becoming weaker during the low estrogenic phase. And this is associated with the changes in gene expression. So this is basically telling us that these 3D genome changes may be functionally important for changes in behavior. We also found around 2,000 differential enhancer promoter interactions. Again, they become more similar to males when the estrogen levels rise. And I'm now just showing you, for instance, this is proestro-specific, like enhancer promoter interactions, and then these are diestro-specific. So just seeing this differential enhancer promoter interaction across the cycle really, for me, is remarkable, right? That you can see such a dynamic change in this. It is remarkable, right? However, of course, the idea would be that 3D genome changes correspond to changes in gene expression. But what is really interesting to us is that only 10% of differentially expressed genes show differential enhancer promoter interaction. So this tells us actually that maybe there are more chromatin changes than changes in gene expression. And so we asked the question, what are these 2,000 differentially 3D genome interactions? What are they important for? So when you do the pathway analysis, you see that the pathways that are changing are associated with brain disorders. And specifically, you see the enrichment of serotonin and anxiety. And this is really remarkable that every time we do pathway analysis, we somehow see serotonin and anxiety showing up. And I want to say one important thing, that this is really specific for the within female comparison. When you compare males to females, you do not find it. So this does show that there is something about serotonin and anxiety that is important when we are talking about these chromatin changes. However, we are thinking if there is some kind of molecular epigenetic priming, where basically there might be more epigenomic changes in response to hormones, and then you just partially change in expression, but you may need actually another stimulus to have actually fully functional change. And I'm going to address this just in a moment. Another question that we can also ask is, like, if you see these changes across the cycle in 3D genome and you take all of them and you do motif analysis, are you seeing the enrichment for estrogen response element or something else? And I'm just going to remind you that with our ATAC-seq data, we actually saw the consistent changes with the membrane-bound receptor. So when you do the 3D genome analysis and you do motif analysis, is this now estrogen response element or something else that is being enriched? And importantly and intriguingly, with the 3D genome data, we always see the enrichment of estrogen response elements. And when we did the confocal analysis, we were able to confirm that indeed ER alpha is present in the nuclei of our neuronal cells in the ventral hippocampus. So we do have both membrane and nuclear estrogen receptors in our cells, that's for sure. So our data actually assume, basically indicating that nuclear estrogen receptor alpha might be important in this 3D genome organization. So we now actually have the evidence from our data that both the membrane-bound and the nuclear-bound pathways are important, right? And I just want to, break at this point, before I give you the last set of data that are more functionally important. So what I show you so far is that there are extensive chromatin organizational changes across the cycle that at least partially overlap with gene expression changes, which means they are important for gene regulation. We are seeing compartmental changes in X chromosome, and this is implying that inactive X chromosome might be breeding across the estrous cycle. This is really interesting to us, and it potentially can be really have a huge implication for the female health. Many chromatin changes are not associated with gene expression, and we believe that this is kind of a molecular priming, that maybe hormones are just changing the epigenome, but you need another stimulus for them to be fully translated into changes in gene expression, behavioral changes. What is important, though, is that differential 3D interaction are enriched for brain disorder relevant pathways and genes. So what we are thinking, if you have another risk factor such as stress, maybe that can actually translate these changes into more functional changes and lead to psychopathology. So we really hope that this can provide a molecular mechanism for increased female vulnerability for anxiety and depression. But of course, the question is all these changes that we are seeing, because people will say, you're seeing that changes in behavior correlate with changes in chromatin, this correlate with changes in gene expression, but how do you know that they are functionally linked? So we use two different approaches to show this. And one approach is actually to replace ovarian hormones. That's one possibility, it's a classical approach. And another approach that I'm going to also highlight is to model the astrocycle stage specific transcriptional program that I'm going to tell you about just in a second. So what we did, we decided to basically remove the ovaries and just give the estradiol and see whether this can actually recapitulate the changes that we are seeing in terms of 3D genome, right? So we have two different groups now. We have all the animals are over-optimized. One group of animals is getting vehicle and one group of animals is getting estrogen, right? And the dose that we used is actually the dose that we showed previously to change behavior, and it was only for 4 hours, right? So what we did, we took the ventral hippocampus, we isolated the nuclei, we did a high C in animals that both, as I said, all the animals are over-optimized. Some animals actually are getting estrogen and another group is getting the vehicle. And then we did a bioinformatic analysis. And remarkably, when we did a complemental analysis, we actually saw that the primary changes that we are seeing are changes in X chromosome. So we were able to reproduce these compartmental changes that are specific to X chromosome. And then when you compare the changes across the cycle, you see around 1,000 differential compartmental region across the cycle, and maybe around 700 when estrogen is given to over-optimized animals, right? The overlap is around 25%. I think this is pretty good. First of all, I already told you that the astrocycle is not only estrogen. And the second thing that is important, that over-optimized animals show already some adaptation. So you do not really expect to be able to completely recapitulate your cycle with the over-optimized animals, right? So you see around 25% of this overlap. We looked also at the enhancer promoter interaction. We found around 15% of the genes that are, shared between the enhancer promoter interaction that are different across the cycle and when you give estrogen to over-optimized animals. And this is just to give you an example of one gene. So this is the changes that we are seeing across the cycle. And in green, you can see what kind of a 3D interaction you can actually reproduce by using estrogen. One interesting thing, though, was when we did the overlap and the motif analysis on these overlapping genes, we were able to see again the, enrichment for EGR1, right? And EGR1 is this novel approach that actually I just want to finish this with, that we want to actually try to model the astrocycle stage by overexpressing the transcription factor that is specific to the proestrus in this case, right? So basically we have EGR1 that is downstream of the estradiol, and we want to actually to check whether the overexpression of this EGR1, which is also seem to be important for both pathways, can actually recapitulate the behavioral and chromatin and gene expression changes that we are seeing across the cycle. So, and again, this was done by Devin and in help with Eduardo Gallo, our actually colleague. And just to remind you, we want to look at the EGR1 because it is induced in proestrus. The EGR1 motif is found in both chromatin and the gene expression data, and it's an immediate early gene that it's a candidate chromatin opener. So Devin actually used the AAV, vectors to an HSYN promoter, which actually will allow you to overexpress EGR1 specifically in neurons. And we also made sure that this is only true in the ventral hippocampus, but not dorsal hippocampus, and that you have a nuclear overexpression. Overexpression is around 6 to 8 fold. So it's not really physiological, but it's also not something crazy, right? And then he looked at the behavioral gene expression and chromatin organization data. So importantly, when you do the overexpression of EGR1, which we consider a pro-astro-specific like transcription factor, Devin was able to recreate the behavioral phenotype that you are seeing with increased estrogen, right? So you don't see changes in behavior that are, in terms of the activity level, are the same in both female groups. But when you overexpress EGR1, specifically in the ventral hippocampal neurons, you are actually seeing less anxiety. And I have to say, importantly, this is also only happening in females, but not in males. So this is a sex-specific phenotype that we were able to show. Then when Devin looked at these OVX animals, the question was like, what is happening molecularly in them when you overexpress EGR1? So what he did, he also did the RNA-seq analysis and was able to show if you overlap the RNA-seq data or differential expressed genes after you overexpress EGR1 in over-optimized animals, and you compare them with the differential expressed genes across the cycle, you see around 33 genes that are overlapping. And when you do the pathway analysis, a lot of these genes are actually important for dendritic spine density and for the synaptic plasticity. And I'm just giving you an example of 2 genes that we know are showing, they are changing across the cycle in terms of chromatin changes and gene expression, and they have the EGR1 motif. And what Devin was able to show, if you overexpress EGR1 in overactive animals, these same gene actually are showing induction in expression. So we see a molecular change. And finally, I want to finish this with this idea. Can EGR1 now that we actually infer from our, ataxic and RNA-seq data in vivo change chromatin? So what Devin did after overexpressing EGR1 in ventral hippocampal neurons and doing ataxic on new positive nuclei, he was actually able to show that indeed, when you see the region that open after overexpression, the majority of region actually have the EGR1 binding site. So indeed, EGR1 seems to be able to open the chromatin in ventral hippocampal neurons. When we look in the females, for instance, in the gene that I already showed you previously, this is a gene that is important for anxiety-related behaviors and also for synaptic plasticity. You see induction of this gene, and this is associated with chromatin changes. So we see that EGR1 overexpression opens the chromatin as we assumed. It actually induces gene expression and it changes behavior. And this is actually sex-specific. So we actually see decrease in expression in males with the same overexpression and no changes in chromatin. So what I want to actually, wrap this up here with this idea that, we were able to actually look across the cycle with the genomic assay, infer the downstream targets of estrogen, and actually in vivo manipulate them and show sex-specific changes in chromatin and behavior, which really I find very remarkable. And I just want to leave you, I promise this is going to be just 1 minute. I just going to leave you with this idea that now what my postdoc Marija is actually trying to do is to look at these same groups of animals in the ventral hippocampus, but do now the single cell analysis. So we do this 10x multiome analysis of both RNA and ataxic data. And so we are able to now see these different clusters of data using this single cell analysis. And if we do the integration of this data, you can see that now in the ventral hippocampus, we are seeing 33 different cell clusters. And I just want to remind you that all the assays that I showed you so far was done on new positive nuclei, right? So we would sort the new positive nuclei, all these red dots that you are seeing here, we would take all these nuclei and then do these assays. So now what we are able to do with a single cell assay is to look at the single cell level and with across all different clusters, and we are going to look across different groups and hopefully actually refine our findings and find a more specific subpopulation of cells that we can also manipulate and further show the targets of estrogen that hopefully at some point we can actually target for treatments as well. So I hope that I actually, persuaded you with this talk that we should take into account the ovarian hormone status and their effect on the epigenome and gene expression and how this can change brain structure and function to actually, link to these sex bias changes of relevance to anxiety and depression. I do believe that hormonal changes is just a part of the puzzle, and we should start looking at the stress. As I mentioned, we have this priming by hormones, and I think the stress can really induce this second stimulus that can actually change gene expression and behavior further and most likely lead to psychopathology in humans. But I really think that by looking and finding this downstream targets of estrogen, we can actually find some new sex-specific target that can be transformational for this treatment of this disorder, because I want to just really emphasize that 2/3 of the patients are women, and we do not take gender or sex into account in the current clinical practice, and this has to change. And of course, this was not done by me. All the experiments were done by my lab members. I would like to particularly thank Devin Roggs, who is graduating very soon. He's done the majority of work that you actually saw here. Leila, also Ivana Jaric, Marija Tikerhoff, who is a new postdoc, and also Fabio Belia, another new postdoc in the lab, and a lot of undergraduate students that actually were also involved in this research. Our colleagues from Einstein, John Greeley, from Texas A&M, Dr. Masako Suzuki, our collaborators from University of Nebraska, Jordan Rowley and Mamta Shukta, our colleagues from Fordham, Eduardo Gallo, Sylvia Fineman, and Haining Chen, and of course, NIH and Brain and Behavior Research Foundation for their financial support. And thank you very much, for your attention. I will be happy to take any questions.
Thank you so much, Marija. This was an amazing talk. So we have some time for a few questions.
And I'm sorry that I went a little bit over time, but
it was great. No problem. So I will start by posing the questions from the audience. So we have a question from Sylvia Enrich. She says, "Hi, Marija, great work. If estrogen seems to cause a general increase in transcription, do you believe the changes in behavior can be linked to changes in single genes or a plethora of genes?"
Yeah, so that's always a question. Like when I give you a single gene, it's just really to simplify things. I think it's always multiple genes, and this is why I prefer looking at something that is upstream of chromatin but downstream of estrogen, right? Because for me, estrogen could really be a good therapeutic agent, but the problem is you have increase in reproductive tumors and cardiovascular risk and all of these things. So the idea would be, can we find something that would be more specific? And I agree, the single genes are just here as examples, but I don't think that any single gene is actually sufficient to explain the complex behavioral phenotypes.
Okay. So we have an additional question from Sylvia. Do you know if there is a compartmentalization of cells that are more modulated by the nuclear versus the membrane activation of ES receptors? Do these cells have different known functions?
Are we now talking specifically about estrogen related or anything?
Yes, and those two branches that you have, one which goes through the transcription factor and the other one which...
So exactly. I think this is all kind of a theoretical working model, to be honest with you. I think we know so little about this, and I agree. There are tools to actually address this. You have, let's say, genetic models where you have only membrane receptor or only nuclear receptors. There are ways of activating only membrane receptor if you use, let's say, the estrogen that is actually bound to BSA or something like that. So I think it's going to be really important for us to, at some point, to dissect this pathways, but my impression from all the data that we have is that both the pathways actually converge at some point.
Okay.
Because I think that's something that makes most sense, because we see membrane bound, like EGR1 seems to be more membrane bound, but then somehow it also overlaps with the ER alpha, nuclear ER alpha path. So I think that there is something actually shared and converging in these cells. And I do want to say, while maybe you're thinking about nuclear estrogen receptor as classical one. In the hippocampus, actually, it's been shown already that membrane-bound receptors are really important for many different behaviors, right? Particularly for these more fast-acting actions.
Okay. Great. Okay, so now we have a question from Rita Figueiredo. She says, "Hi, Marija. Thank you very much for your interesting talk. I was wondering if we assume the pill maintains estrogen." Okay, I think she's talking about birth control pills. Maintains estrogen levels more or less stable. Could it have a protective effect in development of anxiety and depression? And related to this, are there any studies correlating dropping the birth control pill with the onset of anxiety-like behaviors due to more fluctuations in hormones? And lastly, are there any studies on postpartum depression occurrence depending if you took this kind of pills or not?
So these are all important questions. I'll try to be simple. It's not easy, right? First of all, this is such an important area, right? I mean, we have hundreds of millions of menstruating individuals that are actually taking the birth control pills. And we know so little about that. So that's one of the biggest issues in, like, we have, information, like very little information, like, So I was actually looking at some of these review articles, and one of the things is, like, we don't even know, like, whether, like, when you're thinking about the contraceptive pills, what is more important, like regulating endogenous or what these exogenous hormones are doing, right? So these are all great questions. In terms of the risk, it's really interesting because in a majority of cases, stabilizing the hormonal levels should decrease the depression anxiety risk. But let's say that maybe there are up to 10% of people that actually develop anxiety or depression when they start taking pills. And I think this is where the problem is. It's not really about stable level. I think it's really about
fluctuations.
If you get too high, if you get too low, actually, you may be triggering these symptoms, right? So that's one of the problems. In terms of postpartum depression, was the postpartum depression more question about what's happening molecularly, or it was more about
depression, right? If there is any correlation between postpartum depression occurrence, depending if the women took the birth control pill or not before.
So it seems like one important thing across these different, windows when menstruating individuals might be changing hormonally very much, is that if you show sensitivity to hormonal shifts, like let's say you have a PMDD, like premenstrual dysphoric disorder, there is a higher chance that you will see postpartum depression, that there is a higher chance that you will see perimenopausal changes in depression. So there seems to be, that's probably at least in part genetically mediated sensitivity to hormonal changes. Because if you look at all of these individuals that, for instance, have PMDD, they don't have different absolute levels of hormones, right? When you compare them to people that do not have PMDD, but they seem to be more sensitive to these hormonal changes, right? So it's really, there is something about sensitivity to these hormonal changes that I think is important for anxiety and depression, right? And was there another question about this? Because this is a really important area, but as you know, like, it's so little that has been done, and we need to do more to understand this both in humans and in animals.
Yeah. So the other question was regarding dropping off the birth control pill, and if that then can lead to exacerbation of anxiety like behaviors, right?
As I said, like, it depends on the individuals. I think for some individuals this can be stabilized. For some other individuals, if they get triggered by this, actually removing that can actually, so it's not that simple. But in terms of withdrawal, I think the best is PMDD and postpartum depression, when you have like this really like abrupt changes, and this can actually increased risk. So for instance, in postpartum depression, around 20% of birthing people will have postpartum depression. Up to 80% of women will actually, report some mood changes after giving birth, right? So this seems to be very well established, right?
So actually, I have one question regarding to exactly what you are mentioning, right? Because I was thinking about really, what goes on in the cycles, and what you are mentioning regarding that it's not really the absolute, but these kind of fluctuations, right? So I was wondering if there's a way of understanding what is the level of estrogen, which is enough to kind of give you a protective effect in terms of anxiety without, being detrimental for the other functions of estradiol or estrogen in right?
Yeah. So I think that's
there's like a threshold, right?
Yeah. So I think that's a big question. Like also, like when people talk about these studies that show the increased risk for reproductive hormone, it seems that actually these studies were misinterpreted in a way that, like, that the risk is not that much increased. But like, let's say if risk goes from 1% to 1.5, someone said the 50% increase in risk or something like that, so I think that this was basically exaggerated, and now there is this big fear of people to go back to this because, like, now you have to do another natural experiment, right? So I don't know, like, I think there are women that are or people that actually are going through perimenopause, and the hormonal replacement therapy can be very helpful for mood and stuff, like, so I think it's also there is a huge inter-individual variation, in terms of maybe even from one cycle to another, how in a single woman or single menstruating individual.
Exactly. So, and I was also thinking that maybe not at the level of the hormone levels, but also at the level of expression of the receptor. Then if there are changes between individuals, that can also lead to changes in how they react, right?
Yeah, and I think one of the things that we really have to do also to dissect the different receptors, because you have estrogen receptor alpha and beta, you have some G protein coupled receptors that can also. So I think there is also the idea that if you can go a little bit more specific into certain receptor subtype, that can also lead to like a more like a specific action of these hormones as well, right? So yeah, there is so much to be done, but I think what really is exciting is that we are able to go from these genomic studies, find the like a candidate, do overexpression in vivo, change behavior. So it gives me a lot of hope. At some point we are going to find something, and I did not have time to show this, or but we also want to do something in humans too, right? So it's going to be important to actually follow up on this work. So there are a lot of more exciting work actually coming from the lab.
I'm very exciting.
Yeah, I'm also very excited about the single cell data, because this can also give us a very new insight, right? Because sometimes you do like a bulk analysis, and even if we were specific for neuronal cells, right? If there are very specific subpopulations of neurons, you may lose that because there is it's diluted, in this huge differential, like a heterogeneity of neurons by themselves, right? So a lot of new stuff coming from the lab. So please, stay tuned. I would say.
Very, very exciting. So I have some more questions, but maybe we can take them to the Zoom.
Absolutely.
So first of all, thank you so much, Marija, for joining us today. It was an exceptional seminar. So I would like to invite everyone to join us again next week on our next brain-body interaction seminar. And whoever is interested in continuing this discussion, please join us in the Zoom link I just posted on the chat. So just join. And see you soon.
Okay. Thank you.
Bye-bye, everyone.
Bye.